Hey everybody, welcome to lesson 11. Today we're going to be talking about biotechnology and recombinant DNA. So, when most people think of microbes, they probably are thinking about diseases, so how that applies to the medical world, or they might be thinking about, bacteria that's on the surface of things, cleaning your sink out or something like that, that bacterial mat. But we have, through a lot of different discoveries, been able to turn both bacteria and viruses into tools that we use every single day. I've alluded to these a lot, but we're kind of going to dig into them a little bit more and exactly how we use them, especially recombinant DNA, to sort of prop up all of the different uh, pillars of modern day medicine and science. So, really quick, we're going to use some terminology to start. First thing is biotechnology. Biotechnology is encompassing the broad use of an organism or their products to benefit humans. So this is anything from realistically like making cheese to antibiotics to vaccines to biofuels to very, very specific like protein glues and things like that. If we're using different organisms, that means that we're using biotechnology, and that's where it comes from. Bio and technology fused together. And at the core of biotechnology is genetic engineering, where we deliberately modify an organism's genetic material to produce an outcome that we want. A lot of this is done with recombinant DNA technology. So this is combining DNA from different sources into one specific uh, thing, whether that's a plasmid that goes inside of a bacteria or the vectors inside of a virus, whatever it is the DNA from different sources all together, and that's recombinant DNA technology. DNA that's been recombined to produce something new. So it kind of stacks. So you have recombinant DNA, and then above that you have genetic engineering, and then above that you have biotechnology as a whole. So why is all this really important? It's really important because it's how we make modern day biologic medicines. This is the way it happens. You're using some sort of recombinant DNA technology to make cells, make the proteins or metabolites, whatever it is that you want for biotechnology. Now, when we're talking about using recombinant DNA, which is the basis of all this, there's a couple things that really help us out. One is restriction enzymes. Restriction enzymes cut DNA at specific sites, either leaving sticky ends, is what we call them. So these are overhangs. So the DNA matches up from the two ends. So remember, DNA is two-stranded. If you cut, then you can have some sticky ends that then stick to each other and make it so you're not having any degradation of those ends and having the repair mechanisms either put in or take out too many base pairs from your DNA, leading to mutations. So the sticky ends are important in that they prevent mutations. Then we have, of course, DNA ligase comes and seals that DNA together, and that is the simple three steps. So we cut the DNA, make sure it has sticky ends come together, DNA ligase permanently seals them, and this is how we have been doing recombinant DNA for many, many decades now. So that's the general process of how that's done. And then, of course, we want to take that DNA and we want to introduce it into some sort of cell. So there's a couple different ways of doing this. With prokaryotes, you're going to use transformation. So this is uptake. You just have the DNA and then it's uptaken into the prokaryote. This is usually going to be done through some sort of chemical stimulation to kind of open the cell walls and have the uptake happen, or it's going to be used through electric shock, and that's going to allow all the membranes to come apart a little bit and uptake all that DNA. And that's called electroporation. Then you're going to have conjugation. This is direct transfer between different cells, and this typically, isn't really going to happen too much when we're designing the organisms, but it does happen that the organisms will trade their DNA. For eukaryotes, it's a little bit more difficult, and the way we normally do it now is with some sort of viral vector. So you're going to take a virus, strip out all the bad stuff, put in just the part of the DNA that you want to have replicate in the cell, and maybe integrate, if it's a retroviral, into the actual DNA of that cell permanently. You use that viral vector, and then that allows for the DNA to be inserted. And that's the way we get the genetic information in there. But there's also the question of, well, we have all of this recombinant DNA that we've been using over and over again. How do we store that in any way? And there's a couple different ways. There's plasmid libraries. So these are different plasmids that are just frozen, that you extract the plasmid DNA from the bacteria, you freeze them down, and those can be stored. There's phage libraries. So phage libraries are usually you take bacteriophages, those are the viruses that infect bacteria, and they're going to carry the recombinant DNA that you want. These are used very broadly because you can do a lot of robust screening with phage display libraries. And then there's cDNA or complementary DNA, and this is made from processing mRNA, which expresses the genes that we want. And then you kind of take that, flip it around, integrate it into DNA, and make these cDNA libraries. So the libraries are these wonderful information banks that I can call up another scientist who's done experiments, 10 years ago and say, hey, I really want this plasmid from you. And they say, yeah, I have it frozen in the freezer. Let me take it, put it into some bacteria, replicate it a bit, and then ship it off to you. So there's this banking of all of this research that's been done that so it can be built upon over and over and over again. Um, when we are looking at DNA, so say I get that shipment from a scientist, I'm going to want to check and make sure it is the plasmid that we both think it is. And the way we do that is through a PCR. So you're going to run a PCR on that DNA. It's going to show you the band sizes, and you can do restriction enzyme digestion. So those same restriction enzymes that are used to kind of cut and paste DNA together into what we want can be used to disassemble a plasmid. And if you get the right sizes of a PCR on a electrophoresis gel, that can tell you, yes, this is the plasmid we think it is. All these cut sites are where we think they are. Or you can end up with an absolute mess. And it's just like, no, this is not what I thought it was at all. So that's very important. Visualizing proteins, the end product, is also really important because you might get a plasmid that looks correct, but the part that actually codes the protein is very small. It's a small protein, or maybe there's the potential of a mutation in there or a cut site that got cut out. So you want to make the protein and then run it on what we call an SDS page gel. This is a gel that separates the proteins based on size. So it'll go through the gel, and the farther it goes, the smaller it is. So the ones at the top are large, the ones at the bottom are small. So you can look and you can say, well, I'm looking for a 50 kilodalton protein and I'm getting a 42 kilodalton protein. Something's wrong here. So we have all these ways to screen when we do take out of these data banks. And like I mentioned, there's so many medical applications. So you have therapeutic proteins such as insulin, clotting factors that are made for giving to people who have extreme uh, wounds. We have our subunit vaccines are made from proteins that are produced from this recombinant DNA to genetic engineering to biotechnology. So it's ultimately being produced by cells, and that was all done through genetic engineering. A lot of diagnostics use something in this wheelhouse. So a lot of diagnostics use PCRs, which is used in screening all the time. And then a lot of different diagnostics are antibody-based or peptide-based, and they are required to recognize a little bit more complex interactions, and that's another way we can do that. There's also bioremediation. We can engineer microbes and the metabolism in those microbes to break down oil from oil spills or break down plastics. There's actually, I believe it's a microbe in earthworm guts microbiota that can break down styrofoam. So now people are looking at, okay, how do we culture that in mass, expose it to styrofoam, and get rid of styrofoam that way? So very interesting stuff there. Of course, there's gene therapies also that fall into this. So for gene therapy, that means you're taking a viral vector, infecting a person's actual cells, and that infection is going to rewrite part of their host genome. And when that happens, you can do wonderful things like take someone with sickle cell and cure them of all the issues around sickle cell. They'll still have some sickle cell producing cells, but you overcome that by having enough that produce normal red blood cells that you no longer have an issue in your day-to-day life. So that's kind of a miracle cure, right? And there's other things. There's some diseases that cause blindness people are working on. There's a lot of other genetic diseases that people are working on these gene therapy for, and they're really, really cool. But it gets to the core of why this can be somewhat of an issue because what if you're targeting the wrong gene? Now, that's just some very basic overview of everything that is involved with genetic engineering in biotechnology and kind of how microbiology flows into that very clearly. And it's just a very logical next step in what we do as scientists. So I hope that this is very interesting to you. Obviously is very interesting to me. It's what I do all day, every day, for my research life. So uh, there's plenty of examples of cutting edge things that people are doing, and it generally all falls under this umbrella of biotechnology and using microbes really to sort of step into the next era of science and medicine. Hope you have a lot of fun with this lesson. If you have any questions, as always, please reach out and ask. Sorry, the turkeys get to me. Have a fantastic week, and I will see you next week.